Okay, hi everyone. I'm Nick. I am a biology student, and today I'm going to be talking about developmental biology, just giving sort of a basic introduction. So, if you're looking for anything advanced, you are in the wrong spot, but this is generally, say you're taking a class on developmental biology, this is generally going to be sort of what the first lecture would consist of. And I'm just doing this for fun. But if you guys get any sort of use out of it, that's awesome. And if you have any questions that are sort of related to a first time developmental biology class, you can feel free to comment those below, and I'll do my best to answer some of them. And I will admit if I don't know the answer to it, because developmental biology is a field that's constantly moving ahead, and it's something for me that it would be hard for just myself to keep up with. Although many of my professors, there's so much advancement in the field of biology in different areas that it's hard to keep track of all the literature and keep up to date. But yeah, so developmental biology is a really interesting subject in that it is a keystone of how biologists today understand evolution, right? If you look at embryos, how similar they are, and how development proceeds, and how it's not really unique in any species. Of course, there are lots of unique factors, but how similar they are is so remarkable from species to species. But let's talk about what we're going over today. We're going to be talking about sort of general development steps in an embryo, or stages, or how they do it. We're going to go into more detail on that, and then we're going to talk about how cells differentiate from each other, and hopefully we can keep this relatively short. I'm just going to take this off because it's hot in my basement, so apologies for that. But the first thing we're talking about today, as you can see on the board, is how a zygote increases in complexity. And zygote, if you know biology, you know that a zygote is produced when let's use humans for example, a human sperm fuses with a human egg cell, so we get a new individual, right? How do they increase in complexity? Well, number one thing that happens, immediately, well, not immediately, but one of the first steps is cell division, right? So we see that zygote divide, and that is how it increases complexity. Cells break up into more cells. I could do another video on cell division if anyone is interested in that, but at the moment we're just going to stick to our discussion on development, right? So there's multiple rounds of cell division. We're increasing in complexity, right? The next thing that they do is cells take on different identities, right? So I'm just going to write that down. Let's take on different identities. I'm just going to abbreviate different identities. Okay? So through this, we see increasing complexity in the zygote. Cells divide, cells take on different identities. Those are the bullet points, right? So how do we then proceed from our zygote in more detail? How do we go from that to our fully formed organism? There's sort of five main, I don't know whether to call them factors or stages, and they are cleavage, pattern formation, morphogenesis, differentiation, and growth, okay? So firstly, we've got cleavage. Now this is cell division, right? Multiple rounds of cell division. Initially, in most embryos, what we see is once the egg is fertilized, there is multiple rounds of cell division, but without any sort of growth, right? So we're just dividing that egg into smaller and smaller portions before any growth happens. And often there isn't much growth, but we'll get into that. There isn't much growth until later. Another factor is pattern formation. So this is the organization of cells in your body, right? This is the development of a body plan, okay? This is where the body axes are established, right? So the front of the body, the back of the body, the bottom, the top, ventral, dorsal, anterior, posterior. Those are probably the terminology that your professor or teacher would like you to use, ventral, dorsal, anterior, posterior, right? And so obviously this is incredibly important, but one other thing is that it also defines the germ layers. And You've probably seen this in previous biology classes, but germ layers, you've got, right, the ectoderm on the outside. You've also got the, let me get another color. Right, we sort of got the coelom right here. And I need my other color. And just sort of around it, it's right here. So sort of just everything except, so in red here we've got our ectoderm, right? And so what is that going to develop into? Well, in mammals that develops into the epidermis of the skin as well as the nervous system tissue, right? And then in here in green we've got, what is it called? Endoderm. And so endoderm is going to develop into the gut, the liver, and the lungs in mammals. Of course, it's different if you look at insects, but we won't get that in depth because I don't think it's necessary. And so, let me just be more specific. Everything in this green region is the endoderm, okay? So you can pretty much just color it in. All right, and this is an early, I believe this is a gastrula here. So everything in green there, that is going to be the endoderm. And actually, there is some in the middle that's not taken up. So that's going to be a bit of an empty space there, right? Because we are forming the gut with that. So there's going to need to be some trachea, as well as the lungs. And then here we've got the ectoderm. And like I mentioned, the ectoderm is going to be the skin, the epidermis of the skin, as well as the nervous system. And so we can see beginnings of the nervous system right there and the skin around it. And then everything else in here is blue except for this part here that is white. I'm not going to color all of it in because that would take a while. But yeah, and the blue, sorry, I didn't even mention what the blue is. The blue is the mesoderm, right? So this is going to develop into the skeleton, muscles, kidneys, the heart, blood, all that stuff, right? And so if we continue, we move on from that. I am going to talk about these three: pattern formation, morphogenesis, and differentiation are very closely linked, right? All of these processes overlap, but these are particularly linked, and I'll talk about that more in a second. So morphogenesis is the sort of 3D arrangement of the embryo. So if you've ever seen a video on gastrulation, like you can just look up a video of it happening and you'll sort of get the picture. It's sort of, gastrulation is when we go from, and this is not all morphogenesis is, but it's a good example of morphogenesis. Gastrulation is when you go from the blastula to the gastrula. So it's sort of like a linear compact version of cells, and the mesoderm and the endoderm moves inside. You can actually see the cells move up and they create this cavity inside the cell. And it's sort of the first stage of development of the three-dimensional shape of the embryo. And so there's some interesting things to consider in biology when we're talking about plants versus animals in terms of morphogenesis. There's some interesting differences. One of the first differences is that plant cells actually don't move in their morphogenesis. So they don't change locations, whereas, like I just described, the animal cells of the mesoderm and the endoderm, which I never wrote down here, sorry about that, mesoderm and endoderm will move in during gastrulation. Cells do not move in the morphogenesis of plants, which is an interesting thing. They actually control their morphogenesis through cell division and cell expansion. And if you've ever studied any sort of botany, you know that there's many plant hormones that control the ways that plant cells elongate, and there's, a primary, the one that comes to mind is auxin, and they're able to bend towards light. Plant roots are able to direct cell expansion such that they grow downwards in response to gravity. It's actually very interesting stuff. A lot of people aren't that interested in plants, but there's some cool stuff there. There's some cool biology with plants. And another difference is that in animals, morphogenesis ends after embryogenesis. So once the embryo is finished developing, morphogenesis is. But in plants, morphogenesis continues throughout the life of a plant, right? That's why if you've ever had a plant growing on a windowsill, you can actually notice that that plant starts to bend towards the light, right? It's really interesting stuff, and yeah, I think that's about the differences between the two. Just making sure I cover everything. Yeah, so let's move on to differentiation. So differentiation is definitely one you've heard before, and this is where cells develop different identities, right? Muscle cells, cardiac cells, stuff like that, right? And so all these processes here are, linked very closely, but they can also work together to produce many different cells. If you think about cells in like elephants, lions, humans, there are cells that are the same cell type, like muscle cells or skin cells, but they are morphologically very different from each other, right? And that's because of these three factors working together to produce different things. Of course, I can't get too in depth about it at the moment because we're just doing an introduction lecture, but these work together, so like pattern formation can arrange cells differently, morphogenesis can change the three-dimensional structure of them, and then differentiation, say, changes what genes are expressed, and then those working together can create a number of different combinations of different cells, right? And then lastly, we've got growth. And so growth is generally like not super involved at the start, right? Think of humans, for example. There's very little growth, I think, for the first 16 weeks, I think, of fetal development or embryo development. And then once the body plan is established, there is a large amount of growth, obviously, right? And so growth is primarily done through cell division, cell expansion, and there's also the deposition of intracellular materials, which is like bone and stuff like that. And so one other thing people often don't consider is that growth is capable of really changing the appearance of an organism, right? Because the growth is often different depending on which body part you're talking about, right? So again, we go back to the human example. We think of a baby. They often have very large heads before they, very large heads relatively speaking to the rest of their body. And so then when the rest of their body starts to catch up, you can tell that something with a huge head looks different than something with, more proportional body parts, right? Okay, so we sort of got through that. That's the initial portion of what I wanted to mention regarding developmental biology. But we're going to talk a little bit more about differentiation. And so this is more related to how biologists, what's the good word for this? How they term different stages of differentiation, right? And one that I don't have written down here, but I will say off the top is cell fate. Now, what cell fate is when cells are... Okay, let me explain this in a good way. A cell fate is something that a cell will develop into in its normal context, right? So a cell that is positioned on the bicep of your arm, right, where muscle would usually be, its fate is to develop into a muscle cell, right? And so if we think about cells initially in an embryo, they've actually got relatively unlimited fate, right? Especially ones early in an embryo, one that has, they've all got the same genes, but ones that have the same developmental determinants. And developmental determinants are things like mRNA, which is the messenger ribonucleic acids, right? And proteins, and those are the functional aspects. And we will get into how different complements of mRNAs and proteins direct development and how those are segregated. But for now, just know that cell fate is what the cell normally develops into, right? And how do they tell that? They use fate mapping experiments where the scientists, biologists, let's say, will label specific portions of an embryo, right? And how they do that is they, there's lots of different ways, but a big way is to do it through radioactive isotopes, or GFP, something like green fluorescent protein, which is obviously a fluorescent protein, and it's a large protein, so there can be issues with tagging that onto other things, but for the most part, it's been revolutionary, and I believe it did actually earn the person who discovered it a Nobel Prize, but I can't remember their name off the top of my head. But yes, so in order to determine what a cell's normal fate is, biologists will do a fate mapping experiment, and they will label that cell, and they will monitor what it develops into at a later stage, right? So that they can establish what it is. And then the next question they will ask is it specified? Okay, well, what does specified mean? So specified means the cell has all the instructions necessary for it to develop into that cell. So what does that mean? Well, that could be transcription factors, signals, mRNA, right? And so how do they determine that? It's determined through like isolation experiments, right? So they will actually, if they want to know if a region of cells is specified, they will remove it from the embryo, and they will actually culture it in isolation. So this is an isolation experiment. And if the cells develop into what their normal fate is, then they determine they are specified, because once they were placed in an environment that was devoid of any other developmental signals, right, no cells around them to tell them what to do, and if they were still able to produce those cells, they were specified, right? So then the next question they would ask, okay, well, are the cells determined at this stage? Well, what does determined mean? In developmental biology, determination means that the cell is no longer capable of taking on any other fate than its determined cell fate. So how do they test that, right? They test this by, they label the cells that they want to know are determined, right? So again, you're tagging this with an enzyme or a radioactive isotope, right? And then they actually transplant it into a different part of the tissue, right? And they watch it and they see if it develops into its normal cell fate, right? Or does it develop into the fate that is more aligned with the tissues around it. So for example, you've got a group of cells that normally develops into an eye, right? You want to know, okay, at this stage of embryogenesis, are these cells determined to form an eye, right? So you transplant that, and we place it, say, in an embryo that normally forms, sorry about my dogs, normally forms a stomach or something. I apologize. Okay, so sorry, my example was, you've got a region of cells normally develops into an eye, you want to know if it's determined, okay? So we label it again, we transplant it, let's say we transplant it to an area that normally forms the stomach, right? If those cells form a stomach, right, then they were not determined, right? We can't say if they're specified, because maybe they had the instructions to become an eye, but they were influenced by the cells around them to form that stomach. But we can say they're not determined in that case. But if it does form an eye, it's determined. Why? Because presumably the cells around it were sending signals for it to develop into that stomach, but it was incapable of doing that, right? The only cell fate it was able of obtaining is the one that it was determined to have, right? And so differentiation is that irreversible state of it is now a muscle cell. It can't go back. So a cell is differentiated once it takes on that. And sorry, my last example there for determination is known as a transplantation experiment, right? And so that's it. Maybe I'll have an example for you here. Yeah, I think I should. Okay, so we'll just do a little quiz here. So say we've got a group of cells, right? And these cells, they develop into red triangle cells, okay? These cells develop into red triangle cells, okay? But when they're initially in embryogenesis, they're circle cells, okay? So we know that in embryogenesis, they go from circle cells to triangle cells. I want to know at what point these guys are Well, I want to know stuff about their development. I won't say what the conclusion is yet. So we transplant the region of this, and we find, once we culture them in isolation, that they actually do develop into the triangle cells, the normal cell fate. What would you conclude about their development at this stage in embryogenesis? Please feel free to pause the video right now and give it a guess. Okay, so we would determine that these cells are specified. Now, why would we do that? Why aren't they determined? We can't say that they're determined because we have removed these and we're culturing them in isolation, right? So this is different from a transplantation experiment because this is an environment devoid of any signals, right? So if we had performed the same experiment but placed these circle cells into tissue in the embryo that develops into something that's not triangle cells, and they still developed into triangle cells, yes, we could determine that those were determined. Okay, I realized that phrasing was a little bit confusing. But yeah, so this is my first video on any sort of biology topic, so if you found it helpful at all, that's great. If you have any suggestions, please let me know. Yeah, thanks. If you're still watching, I'd be shocked if anybody was watching, but I mean, if you enjoyed it, I'm super glad. Thank you for watching.